This experiment generates fluorescently labeled recombinant Vaccinia viruses through a modular and rapid method based on transient dominant selection. First, create a recombinant vector containing a fluorescent tag surrounded by synthesized minimal regions of homology, as well as markers for metabolic and fluorescent selection. Transfect this recombination vector into infected cells and amplify for recombinant viruses using metabolic selection and fluorescence. Next, remove the metabolic selection to excise the marker cassette and isolate the cells containing recombinant viruses by fluorescent marker selection. Results of viral plaques on cell monolayers allow visualization of individual virus particles that are characteristic of the fluorescently tagged gene.
We developed this method as we wanted to generate recombinant viruses with multiple genetic modifications using a selection method that could be used repeatedly and universally, regardless of the tag. Whilst the principle of transient dominant selection has been described previously, we wanted to apply it on a larger scale and in a modular manner to give us more flexibility in making genomic modifications.
A visual demonstration of this method is critical as recombinant viral plaque screening and isolation can be tricky. Their frequency can be low, and therefore some unique methods are required for efficient recovery of the virus.
To label the viral gene of interest at the N or C terminus of the protein, accordingly identify 150 base pair long flanking regions of homology. Then design an oligonucleotide sequence comprising the 150 base pair left and right arms, separated by a pair of restriction sites of choice. Also flank this region by a second pair of restriction sites that are different from the first pair.